In this video, we're going to look at the brain. You probably noticed, first of all, that we have more brains here than a B-grade zombie movie. The reason is different brains show different things, and I want you to be able to pick up any brain from this class and know the features. I'm going to teach on this one, but please take the time to look at different brains and make sure you can identify all the features that are on your structures list. So let's move these aside and focus solely on this one for now. So the first thing we need to do are identify the different lobes. In the front, this is anatomically correct here. We have our anterior, posterior, superior, inferior, and our lateral and medial. Okay. So in the front, we have our frontal lobe. Here, we have our parietal lobe. In the back, occipital lobe. Here on the side, temporal lobe. The lobes are divided by fissures. Going right down the center, we have the longitudinal fissure. In the back, we have the horizontal fissure. On the side, we have our temporal fissure. And in the back here, dividing our parietal occipital lobes, is our parietal occipital fissure. If we look right down the center, you'll notice this model is color-coded. The actual human brain is not color-coded, but for ease in this classroom, we do have this color marking. This is showing a sulcus that goes right down the center of the brain. That's your central sulcus. This is very important. It divides the front from the back half, the frontal lobe from the parietal lobe. If we look just anterior to the central sulcus, we find the pre-central gyrus. The pre-central gyrus is your primary motor cortex. And if we look behind this, we find our post-central gyrus. That's our primary sensory cortex. I should probably back up a minute and tell you that if you have a bump, it's called a gyrus. Plural would be gyri. If you have a depression, that's called a sulcus, and plural would be sulci. That's true on the cerebrum. On the cerebellum, it's a little bit different. The bumps are called folia, single folium, and the depression are still sulcus or sulci. That's the external portion of the brain. We'll look at the internal portion in just one moment. So what I've done here is I've taken the brain, cut it down the middle. We're looking at a sagittal section. This band of connective tissue here is the corpus callosum. This is a band of heavily myelinated interneurons that go from the two hemispheres and help the two halves communicate. Below that, we see this almond structure. That's our thalamus. And we have a little bump or an adhesion between the two called our interthalamic adhesion. This particular model shows a hole here, and the reason is the other model has a screw that fits into it to put the two models together. Just so you know, the hole is not anatomically correct. There is an adhesion or a bump that you would see here, and you'll see that on the real brain. Below the thalamus, this region is called the hypothalamus, and hanging off the hypothalamus, we have the pituitary gland in the front. In the back, we have the pineal gland. The pineal gland is what helps regulate your sleep. Coming down here, we have the medulla oblongata in front. This is the pons. See that bump that we saw earlier? And this continues down and becomes the spinal cord once it passes the foramen magnum. If we turn this around the back, we're going to see two bumps right here. This is your superior colliculus and your inferior colliculus. We have two bumps on this side, two bumps on the other side. The four bumps together are your corpora quadrigemina. Do you remember in the earlier model of ventricles, we talked about those spaces? Do you see this space here where I can put my probe? Okay, that is your lateral ventricle. So this lateral ventricle is ventricle number 1, or the other side would be ventricle number 2. They go into the space between the two halves of the thalamus. That would be ventricle number 3. Going down this little tube here, that's your cerebral aqueduct. This space here is ventricle number 4, your 4th ventricle. I think that's the majority of the structures. Let me pick up another brain to show you how these can vary from brain model to brain model. This is another brain model. It has exactly the same structures, but because it's a different model, they're depicted in a different way. Here you see the corpus callosum. You can see all those interneurons. You see the giant holes, that's your lateral ventricle. Here is your thalamus, and the space above that is your 3rd ventricle. It's your intrathalamic adhesion. This shows the hypothalamus in front, and this model does show the pituitary gland sitting in front. Remember, this sits in the sella turcica, that little brain, the skull within the skull. On the back here, we have the pineal gland. These dots represent the superior colliculus and then the inferior colliculus. From the 3rd ventricle, we're going to go down the cerebral aqueduct into the 4th ventricle here. The space behind here is your medulla oblongata. In front is your pons, and then as we pass through the foramen magnum, we get into the spinal cord. I'd like to point out the area called the midbrain. This area right in here, that is behind my finger right here, is the midbrain. This includes the cerebral aqueduct and the corpora quadrigeminal. This is a very specific region of the brain. Prior to that, we have the forebrain. After that, we have the hindbrain. But what we have right in this little area in between is the midbrain. One other thing I'd like to show you is that most of the models do show the cranial nerves. This is cranial nerve number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. The interesting thing about this model, as with most models, is on one side, the nerves are very clearly identified by Roman numerals, and on the other side, they are not. Use your head and ask yourself which one we're going to use for testing in the class. It's not going to be the one with the Roman numerals. This is your study one. This is your test side. Just be aware of that.